Hello guys, now I am changing a little bit in my teaching pattern. Okay, so now I will teach you in a point format, like point by point by point. Okay, and if you have any doubt, you can comment below, and this will be in a very short notes type of format. So first one is over AIDS. Okay, it's basically what? Autoimmune deficiency syndrome. Okay, it's causative agents directly replicate in immune system cells. Basically those cells are T helpers. So basically it affects T helpers. Okay, T helper cells. Okay, so as we all know, AIDS is caused by HIV. Okay, don't worry, we will discuss about that also. And it is usually it occurs in immunosuppressed patients. Actually what happened, that HIV virus that caused this AIDS disease, that destroys these T helper cells. Okay, so after that what happens, your immune system will become very much suppressed. So because of that, what happens? AIDS will happen. Okay, so autoimmunodeficiency syndrome occurs in immunosuppressive patients. All right, now what is and what do you mean by immunosuppressed patient? Basically they have multiple infections at a time. Their immune system cannot fight very basic bacteria or viruses also. Okay, so they have multiple infections at a time. Now the second point is basically over Vibrio cholera. Okay, Vibrio cholera, it's a gram negative and coma shaped. Okay, it's a gram negative and coma shaped. And to confirm diagnosis, we do one test, that is motility. In this, basically what we do, we will check the motility. Motility is check firstly. Okay, why motility check? Because it gives a very antique type of mortality. Okay, and that is basically darting motility feature. Okay, darting motility feature. Okay, and how we grow this? It can be grow or cultivate. It can be grow on alkaline 1% peptone water, and then it will give basically colonies like bluish pellicel form. Okay, and it is also positive for, remember this point, it is positive to in ABA diagnostic serum. Okay, it is positive to in ABA diagnostic serum. Basically this Vibrio cholera, it causes a lot of body fluid loss. How? Basically in this, it will cause rice water stool, and because of this rice water stool, the patient can lose 15 liter water loss. 15 liter water loss. It's very huge amount. Okay, and while to confirm diagnosis, motility is check first. And one more thing we can do, that is in this bacteriological research is also done. Okay, so to confirm diagnosis, motility is checked first. Okay, they will show darting motility, and bacteriological research is done. That is all for this second point. Now let's come to our third one. The third one is basically, here we will discuss about what is coli index and coli titer. Okay, so see, if in question they will give you like 500 milliliter. Okay, and in this 500 milliliter they will form 2 colonies, something like this. Okay, 2 colonies. Okay, now find out the coli index and coli titer. Now see, first of all we have to convert this 500 ml into SI unit, and the SI unit is 1 liter. So 1 liter is basically 1000 milliliter. Okay, so 1000 milliliter. Okay, so how can we convert 500 ml into 1000? Basically multiply by 2. So obviously on the another side also we will do multiply by 2. It will be 4 colonies. It's very simple concept. Okay, like in 500 ml, there will be 2 colonies. So how much will be in 1000 ml? There will be 4 colonies. So these 4 colonies, they are coli index. It is coli index. Coli index means amount of E. coli in 1 liter. Okay, that is coli index. Amount of E. coli in 1 liter. Okay, now what is coli titer? Coli titer. So in coli titer, basically what we do, we will divide this 1000 milliliter to 4. So it will be 250. How much? 250. And this 250 is coli titer. So this is coli titer and this is coli index. Okay, now let's come after. I hope everything is clear from this part. Now let's come to another part, that is our typhoid diagnosed. Okay, typhoid diagnosis. So diagnosticum is carried out in case of typhoid suspicion. Okay, so first thing which we can do basically diagnostum. So first thing we can do, diagnose, it is passive. We can do that passive or indirect heme agglutination, indirect heme agglutination test. Okay, and as like to final, like the final identification. So the final identification test, here we can use the zero identification method. Zero identification. Okay, so basically what is the zero identification? In this we will find the antibodies in the serum. Okay, we will find the antibodies in the serum of that patient, obviously. So in what ratio, what will be the ratio, like the typhoid is confirmed or absent? So what will be the ratio? So the ratio is like this. If it's 1:20, then repeat test after 1 week. Okay, if the ratio is like this, 1:80, then the person is carrier or that patient is carrier. If the ratio is 1:100, that means diagnosis confirmed. Confirmed. CONFI confirmed. FIRMED confirmed. Okay. And yeah, one more thing, in this this passive or indirect hemagglutination test, what we will check basically in this test? In this test we will check VI antigen. Okay, VI antigen. Now let's come to another point, that is basically mass prophylaxis in diphtheria. Mass prophylaxis in diphtheria. So what is mass prophylaxis? Whenever you have to treat or you have to save a very huge number of population, like in a school, okay, in a office, or okay, at that time, at very mass prophylaxis level in diphtheria, we will give. So what we will give? We will give anatoxin or diphtheria toxoid to do the mass prophylaxis, okay, in diphtheria. Okay, so don't worry about diphtheria, we will discuss about diphtheria little bit later on in this video only. So mass prophylaxis in diphtheria, so given is in this we give anatoxin or diphtheria toxoid. Remember this point. Okay, so the another one is immunofluorescence test. Okay, so in this immunofluorescence test, it require fluorescence microscope. Okay, and this immunofluorescence is used to increase information mativeness of microorganisms. Okay, so basically they will be look much more clear, or much more their every part or everything inside that organism will very much in that microorganism will be very much visible. Okay, now let's come to over that phage typing or that phage diagnosis. Okay, is done in basically this phage typing or phage diagnosis is done in basically first one is Staphylococcus aureus, and the another one is Shigella species. Okay, so this is Staphylococcus aureus. Let me tell you something about this. This is basically gram positive, and in this the selective media is glucose peptone broth. What is the selective media means? Basically the selective here means, see, basically it is used to isolate this Staphylococcus aureus from another species. Okay, and yeah, one more feature, it forms, whenever it will form its colonies, it will form grape like clusters. Okay, now this Shigella species, okay, to identify this we will do agglutination test. Okay, and to confirm the test, okay, to confirm the test, okay, we will do bacteriological diagnosis is done. Okay, now another point is, see, what is the difference between active and passive immunity? Okay, so as we know, active immunity is when like your own body's immune system will form antibodies, or it will form antibodies against any antigen in your own body. Okay, so at that time it's active immunity. But if antibody, AB means antibody, is given to a patient, then immunity is what type? Then that immunity developed in that patient body is passive. So basically here we are doing what? We are giving antibodies from the outside. Okay, then it's passive immunity. Let's discuss about brucellosis. Okay, so we are discussing about brucellosis allergy. Okay, allergy test. Brucellosis allergy test. Okay, first of all, watch what type of test it is. Okay, so this is here, it's give type 4 hypersensitivity. Okay, why type 4 hypersensitivity? Because at the site of injection, the T cells found at the site of injection. Okay, and this allergy test have one more name. Its name is basically, as you know, Burnett's intracutaneous allergy test to detect brucellin. Okay, and how this brucellosis is like can spread? Okay, so it is spread through cattle, sheep, pigs, unpasteurized milk, and like over soft cheese, okay, and we can also do one more test to detect this brucellovirus, that is known as, you can write that, okay, here, WRIGHT, Wright's test. Okay, Wright's test for diagnosis. And this allergy type of test name is Burnett's intracutaneous allergy test. Okay, and from type 4 hypersensitivity, I want to tell you one more thing that is not related to this brucella. Okay, see, in type 4 hypersensitivity, there is one more thing, as we just discussed in our previous videos of hypersensitivity reaction, graft rejection is also under type 4 hypersensitivity. Like if someone's kidney is replaced to like, okay, from another person's kidney, so and your immune system is reject that graft, okay, so graft rejection is also under type 4 hypersensitivity. Now do you know how we can sterilize a sugar containing media? So for that, remember this: sugar containing media is sterilized 30 minutes, like 30 minutes and 3 days in succession. Okay, succession. Now, as I told you, we will discuss about diphtheria a little bit. So first of all, what will be the treatment? Another point, okay. Treatment of diphtheria. Treatment of diphtheria. In this, we can use antitoxic serum. Okay, and what will the unit of this antitoxic serum? Remember this thing. So the unit is international. So the unit is international. And one more thing, diphtheria, this can do phage conversion. This can do phage conversion. Okay, so one more point, it won't FORM, it won't form exotoxin. It won't form exotoxins. Okay, and what will the test? So the test for this diphtheria, we can do like ELK test, ELK, ELK test. Okay, ELK test, SHIK test, SHIK test, and very much important, precipitation line test. Precipitation line test. Okay, and what will the culture? Okay, to grow this. So the culture is like blood cystine, tellurite agar, or precipitation agar. Okay, and what will be the I want to tell you one more point about this medium. Okay, so media or medium. So the Loeffler, EFFLER, Loeffler medium. Okay, so that's all from this short note part. Okay, so we will continue these short notes in our next video. So if you have any doubt, you can comment below. Thank you.